Greetings and welcome to Chapter 15. In Chapter 15, we talk about the senses. So let's do a little overview of sensory receptors and sensations. We covered a little bit of this in Chapter 14. So a sensory receptor is able to detect and convert any stimuli originating from both of the internal and external environments into what we call nerve impulses. This is commonly referred to as sensory transduction. Some of the sensory receptors are modified neurons and others are specialized cells closely associated with neurons. Exteroceptors are sensory receptors that detect stimuli from the outside of the body, whereas interoceptors receive stimuli from the inside of the body. Examples of interoceptors and baroreceptors, which are also called pressoreceptors, kind of hard to say sometimes. These respond to changes in the blood pressure. Osmoreceptors monitor the body's water-salt balance, and chemoreceptors are receptors that monitor the pH of the blood. And if you remember, we talked a little bit about osmoreceptors in an earlier chapter. So the sensory receptors in all of us are classified into four categories: chemoreceptor, photoreceptors, mechanoreceptors, and thermoreceptors. Chemoreceptors respond to chemical substances in the immediate vicinity, meaning in the immediate vicinity of it. Nociceptors are what we refer to as pain receptors and are a type of chemoreceptor. Then we have the photoreceptors that respond to light energy. And as we know, when we walk out of a dark room, we always shade our eyes because of the light being our eyes being too sensitive to the light. So our eyes contain what's called photoreceptors that are sensitive to light rays and thereby will provide us with a sense of vision. Stimulation of these receptors, known as rod cells, result in what we see as black and white. Stimulation of the photoreceptors, called cones or cone cells, result in our color vision. Mechanoreceptors are stimulated by mechanical forces, which most often result in a pressure of some sort. They respond to heat, cold. They play a major role in the regulation of the internal body temperature. So how does sensation occur? Well, when there's a stimulation, sensory receptors initiate nerve signals that are transmitted to the spinal cord or to the brain. Sensation occurs when the nerve impulses reach the cerebral cortex. And before sensory receptors initiate that nerve signal, they carry out integration, which is the summing of signals. And then we have sensory adaptation, which is a decrease in response to that stimulus. Getting back to the senses, we have somatic senses. These are receptors associated with skin, with muscles, our joints, and viscera. They send nerve impulses via the spinal cord to the cerebral cortex, and these generally are sensory receptors categorized as proprioceptors, which are involved in the reflex actions that maintain muscle tone and the body's equilibrium and posture. Cutaneous receptors And we know cutane means skin. So the dermis of the skin contains these receptors, and they are sensitive to touch, pressure, pain, and even temperature. 3 types of receptors detect touch. You have your Meissner corpuscles, your Merkel discs, and your free nerve endings. Pacian corpuscles, Rufian endings, and Krauss end bulbs are the ones that detect the pressure. Temperature receptors and pain receptors are what we call free nerve endings. So, like the skin, many of the internal organs have pain receptors. We also call these nociceptors, which are sensitive to chemicals that are released by any of the tissues that have been damaged. And sometimes there's a stimulation of internal pain receptors that we perceive as coming from the skin. This really is just referred pain. It's referred from another area. Then we have the sense of taste and smell. Taste and smell are chemical senses that are sensitive to molecules that we have in food and in the air that we breathe. Chemoreceptors in the carotid arteries and the aorta are sensitive to the pH of the blood. So in other words, whether it's acidic or alkalinic. Chemoreceptors are divided into receptors that respond to distance or direct stimuli. So the sense of taste in adult humans, we have approximately 4,000 taste buds located primarily on the tongue. They occur along the walls of the papilla. Papilla on the tongue contain taste buds sensitive to sweet, sour, salty, bitter, and of course our favorite, umami. And just a little trivia, that's Japanese, it means savory or delicious. These taste cells are microvilli, are in the microvilli, and they possess receptor proteins for certain molecules. When the molecules bind with the receptor proteins and nerve signals are generated, sent to the brain where the sensation of taste occurs. In food, or in a food, it can stimulate more than one of these types of tastes. The brain will appear to survey the overall pattern of the incoming sensory impulse and then make a weighted average of their taste as it perceives from the messages. So how does the brain taste information? So the taste buds open at a taste pore. They have little supporting cells and a number of elongated taste cells that end in microvilli. When the molecules bind to those receptor proteins of the microvilli, then that signal is generated in sensory nerve fibers that go to the brain. Signals reach the gustatory or taste cortex, and that's located primarily in the parietal lobe, which, if you remember, the parietal lobes are right here, where they're interpreted as particular tastes. Then we have the sense of smell. Did you know that approximately 80 to 90% of what we perceive as taste is actually what we smell? The sense of smell depends on olfactory cells located in the olfactory epithelia that are high in the roof of the nasal cavity. Olfactory cells are modified neurons. Each one of them bears receptor proteins for odor molecules. Now, this is how the brain receives odor information. So an odor contains a lot of odor molecules which activate a characteristic combination of receptor proteins. And that odor signature in the olfactory bulb is determined by which neurons are stimulated. The neurons that are stimulated communicate this information through the olfactory tract to the olfactory areas of the cerebral cortex. The number of olfactory cells decline with age. As we get older, our sense of taste and smell decreases. Vision. Well, how do we sense vision? Vision uses the eyes in the brain. The integration of stimuli occurs in the eyes before the nerve signals are sent to the brain. About a third of the cerebral cortex takes part in processing all of our visual information, everything that we see. So a whole third of it occurs in the cerebral cortex. Let's look at the anatomy of the eye a little bit. So the eye really is an elongated sphere about 2.5 centimeters in diameter. It has three layers: the sclera, the choroid, and the retina. The outer layer is made up of white fibrous sclera, and then there's the transparent cornea, which is made of collagen fibers. It's also known as the window of the eye. The choroid's the thin middle coat, and it has an extensive blood supply, and its dark pigment absorbs stray light rays that the photoreceptors have not resorbed. This helps in our visual acuity. You have the iris, which regulates the size of the pupil. There's a hole in the center of the iris through which light enters the eye. And behind the iris, through which the light enters, you have the choroid, which thickens and forms the circular ciliary body. And that contains the ciliary muscle. Ciliary muscle controls the shape of the lens for near and far vision. And the lens is attached to the ciliary body by what we call suspensory ligaments, and it divides the eye into two compartments, anterior and posterior. The anterior is filled with a clear, watery fluid called the aqueous humor, and the small amount of the aqueous humor is continually produced each day. And that leaves the anterior compartment through tiny ducts. The retina is located at the posterior or back compartment of the eye, and it's filled with clear gelatinous material called vitreous humor. It holds the retina in place and supports the lenses. It contains the photoreceptors called rod cells and cone cells. That's our black and white and color receptors. These are sensitive to light, but they don't detect the color itself. Therefore, at night or in a darkened room, you only see shades of gray. So the cones that require bright light are sensitive to different light waves, or different wavelengths of light. And this gives us the ability to distinguish colors. What does the lens do? Well, the cornea is assisted by the lens, and the humors focuses images on the retina. This is where visual accommodation occurs for close vision. During the visual accommodation, the lens changes its shape to bring the image into focus. So it stretches out like this and back in until it gets it focused. There's a pathway for vision to the brain. It begins once light has been focused on and by the photoreceptors in the retina. Some integration occurs in the retina, where the nerve signals begin before the optic nerve itself transmits them to the brain. Function of the photoreceptors in the eye, you have two types. The outer segment of the rods and cones, which stacks of membranous discs which contain visual pigments. And in the rods, the membrane of each disc contains rhodopsin. Rhodopsin is a complex chemical containing protein opsin and pigment retinol. When rhodopsin absorbs light energy, it splits, releasing retinal, which sets in motion this little cascade of reactions that causes the ion channels in the plasma membrane to close off. And thereafter, nerve signals shoot to the brain. The cones are located primarily in what we call the fovea and are activated by bright light. They detect every little fine detail and the color of an object. So they're detecting every single last bit, so that the color vision, which depends on 3 types of cones. So we have these cones, they contain a pigment called B for blue, G for green, and R for red pigments. Each pigment is made up of retinal and opsin. There's a slight difference in the opsin structure in each one of them. Excuse me. This accounts for their individual absorption patterns. Now, the retina has 3 layers of neurons. The rod and cones synapse with the bipolar cells, and in turn synapse with ganglion cells that converge to form the optic nerve. And this transmits information to the visual cortex. As these signals pass from one layer to the next, integration occurs because each layer contains fewer cells than the previous layer. What are some abnormalities of the eye? What can we have? We can have color blindness. Color blindness and changes in physical shape of the eye are 2 of the most common visual abnormalities. And there are several forms of color blindness, all of which are attributed to a genetic mutation. We have types of vision. We have nearsightedness. Excuse me. Nearsighted people can see close objects better than they see objects at a distance. The shape of the eye in these people is elongated eyeballs, and they detect and can be corrected with concave lenses. Farsighted individuals see distant objects better than they see close objects. And the shape of the eye in the individual is seemingly shortened. The defect, we can correct that also with concave lenses. So concave, if you remember, concave is indented. Astigmatism is corrected by unevenly ground lenses because our vision is uneven. Hearing. Talked about the eye, we got to talk about the ear. So the ear's got 2 sensory functions, hearing and giving us balance in our equilibrium. Sensory receptors for these are located in the inner ear. Each one consists of hair cells with stereocilia, which are long, stiff microvilli, and these are sensitive to mechanical stimulation. They act as mechanoreceptors. The ear is divided into 3 parts. You have the outer, the middle, and the inner. The outer ear consists of the pinna and the auditory canal that directs the sound waves to the middle ear. In other words, that's the opening in that little tunnel to the inner ear that sound travels through. And the middle ear begins with the tympanic membrane. And at the tympanic membrane, we have the ossicles, which are the smallest bones in the body, the malleus, the incus, and the stapes. The malleus is attached to the tympanic membrane, and the stapes is attached to the oval window, which is covered by a membrane. The fluid-filled inner ear contains the cochlea, the vestibule, and the semicircular canals. So how does sound make its way to the brain? It begins in the auditory canal. And after that, hearing requires the other parts of the ear and the cochlear nerve and the brain. So it begins when waves enter the auditory canal. Vibrations are amplified by the tympanic membrane. The three bones of the middle ear pick up that motion. And this is how the pressure is passed to the fluid within the cochlea. Vibrations set up pressure waves within the cochlea, which contain the spiral organ or organ of Corti, consisting of hair cells whose stereocilia are embedded within the tectorial membrane. In the auditory cortex, they're interpreted as sound. And we have the sense of equilibrium, which is one of the functions of the ear. So the vestibular nerve and proprioceptors are necessary for maintaining our equilibrium. The vestibular nerve originates in the semicircular canals, saccule and utricle. It takes nerve cells signals to the brain stem and the cerebellum. Through its communication with the brain, the vestibular nerve helps achieve equilibrium or that balance. But other structures of the body are also involved. Vision, for instance, if it's available, usually provides an extremely helpful input that the brain can act upon. Well, that has been our chapter 15. I hope you've enjoyed the lecture, and please be mindful to get your work done for this week, and I will see you next week. Have a good day.